Okay, welcome to Let's Get to the Marks, going through the GCSE Combined Science Trilogy 2021 papers. An hour and 15 minutes. You got 6 questions. I'm just going through this one straight through. So, you know the coup. Pause the video, have a go, and unpause and watch the answers. All right, let's get straight into it. Plants absorb light for photosynthesis. Which is the correct equation for photosynthesis? And it's worth a mark. So you want to be thinking to yourself, what is it that plants take in? Do they take in C6H12O6? That's glucose. No, they don't. And they don't take in oxygen. Well, not for photosynthesis, they don't anyway. They do take in carbon dioxide and water, don't they? And they make sugar and oxygen. So this is looking good. Let's just check the others. Water and oxygen, no. And that's oxygen and carbon dioxide, both gases, saying they make sugar and water, no. So there we go. 1 mark. Let's go. Don't forget to press pause on the video once I start the question, and then have a go on a bit of paper. The student investigated the effect of light intensity on the rate of photosynthesis. Figure 1 shows the apparatus. You can see they've got a lamp here. You can see they're controlling the distance. They might be changing it. We'll find out in a second. Using a ruler, because we got the pondweed set up at 10 centimeters from the light source, and we've got bubbles of a gas coming out. This is the method used. Set up the apparatus as shown in figure 1. Place the pondweed 10 centimeters away from the lamp. Switch on the lamp. Record the number of bubbles of gas produced in 5 minutes. Repeat steps 2 to 4 with pondweed at different distances from the lamp. So we can tell what our independent variable is. What was the independent variable in this investigation? Tick 1 box, 1 mark. And I've said this before many times. When you get a method, the independent variable is normally the last step of the method because it's what they change. And they normally talk about repeat steps 2 to 4 with pondweed at different distances. So you'll normally find the IV in the last step of the method. And so that means the independent variable is the distance of the pondweed from the lamp, and not these ones because they didn't say repeat by changing the bubbles, which you can't do anyway, or time taken to collect the gas. They didn't say any of that. They said repeat at different distances. Okay. The lamp gets warm when it's on. This causes the temperature of the water to increase. 1.3. Explain how an increase in temperature would affect the results of this investigation. So there's 2 angles you can go down here. You should pause the video and have a go. Okay, I'm going to go through the 2 different kind of explanations. You can tangle them both in together, but obviously if we allow temperature to increase, temperature is a limiting factor. So this is one of the things that we must control. You've got to control all your limiting factors except for the one you're changing. They're changing light intensity, so they want to keep CO2 and temp the same in this because they're the other 2 limiting factors. Temperature is a limiting factor. Increasing temperature increases the kinetic energy, making enzymes work better and faster, so affecting rate of photosynthesis. So we could go down that route that, look, if we let it get warm, the plant's actually going to do photosynthesis faster because the enzymes involved in photosynthesis are going to work better. It's closer to the optimum temperature. I'll probably want to use the word optimum here, which I haven't used there. Closer to the optimum temperature. I might want to say there's more collisions between the substrate and the active site of the enzyme, meaning that we're going to get photosynthesis faster. But my preferred answer is probably the second angle of attack, which is too high temperature. What does a very high temperature do to enzymes? Too high temperature denatures enzymes. Their active site changes shape, will affect the rate of photosynthesis. So I'm just explaining what denaturing is, that temperature is linked to the denaturing of enzymes, what that means, and that will affect the rate of photosynthesis. So obviously, if the active site changes shape, it can no longer bind with the substrate, and that is going to basically kibosh the photosynthesis. Enzymes actually, interestingly, control every biological reaction. So there isn't a reaction in your body that's happening that isn't linked with enzymes. 1.4 suggests one way the investigation could be improved so the temperature of the water does not increase. What's the piece of equipment we can use to keep the temperature stable? And you should know the name. If you don't, this is something to put on a flashcard. It is a water bath. So rather than just write water bath, because you might get an examiner who says use a water bath. It's a little bit better to say that. Use a water bath. Now you can use an electric water bath is the best one to use. An electric water bath because you just plug it in and you set the temperature. It has its own thermostat, so you can set it at 20 degrees, 30 degrees, 40 degrees. But you can make your own water bath using a beaker, water, a thermometer, and a Bunsen burner. But it's a little bit more difficult because you constantly got to have one of your team taking the Bunsen burner on and off to keep the water at the same temperature. So that's a lot more difficult than using an electric water bath. Let's zing down to 1.5. Suggest two improvements to the investigation so the results would be more valid. Do not refer to controlling the temperature because we've already mentioned that. So this is important actually because this is talking about validity. Now, what does the word valid mean? Whenever you see valid in GCSE science, it's talking about your control variables. What haven't you kept the same that could affect the rate of reaction or could affect the experiment? So when we zing up to the method, they did say they're going to change the distance of the light. That's fine. That's the independent variable. And we know they didn't control the temperature and they should have done, but they've said don't mention that because we've talked about it already. So there's one other limiting factor, not temperature, not light, it's carbon dioxide. So you need to know how to keep carbon dioxide the same. And then if you come up here, there's something really crusty that's not very valid about what they're doing here. Look at these bubbles and look at the sizes of these bubbles. If I count these bubbles, there's three bubbles. But what if I have three bubbles that are like that? It's going to go down in my results that those three bubbles are the same as those three. And of course, they're not. They're different sizes. So we need to measure the volume of oxygen or the volume of gas being given off. And we can do that with a measuring cylinder or with a gas syringe. That's going to improve the validity of the investigation. So let's go down here. So I would say measure the volume of gas using a gas syringe. Okay, I prefer the gas syringe, but you can use a delivery tube and a measuring cylinder filled with water turned upside down. I would also say control, or it's basically control, but use the same mass of sodium, hydrogen, can't spell, hydrogen, carbonate each time. Now, you may not have heard of this sodium, hydrogen, carbonate. This substance releases carbon dioxide when it mixes with water. And so by controlling the mass or the concentration of sodium hydrogen carbonate in your experiment, you can ensure that every plant you use gets the equal amount of carbon dioxide. So one doesn't have an advantage over another. You can also repeat this investigation, but this time keep the light intensity the same, the temperature the same, and then investigate the effect of carbon dioxide by changing the mass of sodium hydrogen carbonate each time. Anywho, if you don't know that, get that on a revision card because that's probably one of the harder bits of the limiting factors, how to produce carbon dioxide and how to control it. One shows the results. Table one, distance of the pondweed from the lamp, 10, 20, 30, 40, 50. Number of bubbles produced, 120, 56, 31, 16, 10. So we've got a set of results here. Calculate the rate of photosynthesis when the pondweed was 40 centimeters from the lamp. Give the rate of photosynthesis as the number of bubbles produced per minute. So this looks nice and easy. Let's take that down. This is the number of bubbles produced in 5 minutes. So we need to bear that in mind. Let's go. When the distance was 40 over here, it produced 16 bubbles in 5 mins, right? So how many did it produce in 1 minute? So we need to do 16 divided by 5. And guess what? I'm so crusty at maths, I'm going to have to bust out the calculator for that. 16 divided by 5. And that is 3.2 bubbles of gas produced per minute. She's having the night wee wees at the moment. She's only 2.5. 1.7, give one conclusion that can be made from table one. So if we bust back up to table one, what is one conclusion we can make? What can we say about as the distance increases, as the distance of the pond weed from the lamp increases, what happens to the number of bubbles? They go down. So we can say the conclusion we can make, which is just a simple description. Don't try and add science in. Just keep it simple, keep it straightforward for that mark. So as distance increases, number of gas bubbles decreases. And that's just a straightforward, simple trend that we can spot. Okay, what we need to do here is plot the data from the table. So our first one, distance is 10, really clear and easy to see on this table, and 120. So we're going to put our first dot there. We're then going to go to the next distance, which is 20, and we're going to go up to 56. Now becomes the problem. We've got to work out what these are. Remember, there are 10 squares here, but there is a value of 20. So we want to find out how much is each square worth. So we do 20 divided by 10 equals 2. Each square is worth 2 points. So there's 50, and 56 is going to be 3 up. 3 times 2 is 6. So 56 is there. I need to go over to the right bit, which is 20 there. And there we go. You're allowed to be half a mini square out. Anywho, let's move on. 30 goes up to 31. So, down to 31. So 30 is there. 31 is going to be right bang in the middle of a square. So don't worry too much. That one is going to be hard. 40 is 16. Now this is 10, so I need to go 3 up again, and because each mini square is worth 2 on the way up, and then 50 over here is worth 10, so it's going to be there. And then it asks me, plot the data from table 1 on figure 2 and draw a line of best fit. Now, some of you might be thinking, hey, do I do like a straight line with my ruler through as many points as possible? You do sometimes, but not if there seems to be a natural curve. Look where we're going. If you kind of put your pen through, it looks like there's a natural curve. So what you want to do is not as bad as mine. You don't want to take your pen off. Notice no matter how bad I'm doing, I'm not taking my pencil off. Ah, nearly through all the points. So you don't want to take your pencil off. What you don't want to have happen is this. This is not what they want you to do. They don't want you to join up the dots. So don't do that. They want a smooth curve, which is difficult to do. When you're doing it on the computer, smooth curve through all the points in one stroke. Okay, you might want to practice that, scribble it down. After 5 minutes, you'll get the hang. Okay, so that's going to score us 3 lovely marks for the plots and the line of best fit. So what we're going to do next, I'm just going to scroll down here, predict the number of bubbles that would be produced in 5 minutes if the pondweed was 60 centimeters. So we want to come back up, see if I can get this place back. Boom, I can. So what you're going to do now is draw a little dot here and go, right, what's the number of bubbles? I'm going to draw a line across. So essentially, we're plotting as if there was one done at 60 and it would be there. So I predict at 60 centimeters, the number of bubbles would be 8. They probably give you a range from like 6 to 8 or 6 to 9 or something like that in the mark scheme. So that's totally cool. Right, let's go down. I'm going to put that down as 8. There'll be a bit of a range there, but you've just got to use your graph, and the examiner will check your graph. And if it's where your graph goes, they'll see if you can read your own graph. So go with however you've drawn it. The examiner will give you that mark. Even if your line of best fit is wrong and you don't get the mark for your line of best fit, you'll still get the mark for making that prediction. Now, we couldn't make that prediction if I'd made a straight line. So that's further confirms we should have done a curve. Okay, we're on question 2 now. Describe how to test a sample of food for protein, starch, and sugar. Give the colors that would be seen if the food sample contained protein, starch, and sugar. What a lovely 6 marker. This is just content knowledge. But what I do find with a lot of students that I teach is that their food tests are it's pretty easy, the food test. It's just some revision cards. Learn it before the exams, off by heart. But it's surprising. I'd say 70% of students don't know their colors and don't know the food test, even though it's just a little bit of memory recall. So describe how to test the sample for protein, starch, and sugar, and what would be the positive result. So let's start with a protein. So for protein, or I'm going to say for all samples, this is something that we probably normally miss out because we can't be bothered, but for all samples, mush um, up the food um, and mix with water. We could then say built leave for 5 minutes, then filter off the solution and test the solution. So essentially what we're doing is we're mashing up the food and crushing it all up to get the juice out the food, get the protein, the starch or the sugar out the food and mix it with water so it can actually be put in a well and tested. Because sometimes food has skin and peel on it and the test isn't going to work like that. Okay. You don't necessarily get marks for that, but sometimes that could be on the mark scheme. So it really looks like it's 2 marks for protein, 2 marks for starch, 2 marks for sugar. And that's probably, you probably won't get a mark for a little intro there. Okay. So for protein, I've got some tips on how to remember it. So for protein, we use the Biuret's test. It's actually called Biuret's reagent, but so use Biuret's reagent, Biuret's solution, doesn't really matter. So we're going to add Biuret's. Um, it will turn purple if protein is present. Now you're allowed to say pink, violet, or purple because those colors are all very similar and kind of subjective to the eyes, aren't they? Oh, that's purple. No, no, I reckon that's violet. So you're allowed to say that. The reason why do I go for purple, sorry, why I go for purple is because P in protein begins with the letter P, and so does the color purple. You could say pink as well, but both of those begin with the letter P. So I like that. Protein goes purple. So it's a good way to remember it. Something else to remember about Biuret's, but you don't really need to say it for either of these, but Biuret's begins with a B and it is blue at the start. So that's another little letter kind of memorization thing. It is blue, but Biuret's is blue but with protein goes purple. So you can use the letters to remember the test. Okay, that's going to get you 2 marks. Bish bash. Now let's look at the starch. So for starch, um, use iodine. It goes from orange. Now they'll give you the mark if you say that iodine is orange or brown. Kind of looks like henna when you get henna tattoos. So it goes from orange or brown to blue slash black. So it goes dark when it mixes with starch. Now, I haven't got anything funky for that, but funnily enough, students tend to remember that one. I think they've been doing it since year 7 and using it a lot. So students tend to remember this. They don't have an issue with this. So you get 2 marks for that, saying use iodine. Orange, brown to blue, black. Now the sugar is slightly more complicated. So for sugar, we use something called Benedict's test. So, or Benedict's reagent. We can say you can say Benedict, do the Benedict's test, do the Benedict's. Now, just like Biuret's handily for us, Benedict's begins with a B and it's blue. So if you want to say what color it is at the start, normally they don't give you marks for saying what color it is at the start. They want to know the color it goes with a positive result. But anywho, it's blue and it begins with a B. But this time, use Benedict's reagent, warm it. So this is the extra thing. It's the only one you warm. So you're going to warm it. It's going to get you an extra mark, potentially. You're not going to get 7. The maximum you can get is 6. So use Benedict's reagent, warm it. It turns from blue to a red precipitate. Well, what that means is you get a red solid hanging out in the blue stuff. Now, there is a little bit more I'm going to talk about with the Benedict's test. I know you might say, hey, is there any letters? There aren't really any letters you can remember anything from. But if you can know that Biuret's is blue, um, yeah, and that the P purple, but who knows? Right, let's go. Now, so what you're getting with the Benedict's test, it's blue and then it goes red. But although you don't have to say this here, the Benedict's test does go orange. It does go green. This test does go green if there's hardly any sugar in there. It does go like yellow and then orange before going red. So it's kind of, we call the Benedict's test is semi qualitative. What does that mean? If it was quantitative, you could say, oh, there's 15% sugar in this food, or there's 25%. It's not quantitative, but you can kind of say, well, this one went yellow and that food went red. That one, the red one, has got more sugar than the one that went yellow. So we call it semi-qualitative. You can kind of compare different foods, but you have no idea of how much sugar that actually is. So it's not a quantitative test like you get with a glucose meter or like you get with something digital. This is just a color change. So it's semi-qualitative. It gives you like sorry, it's semi-quantitative is better to say it because it kind of gives, it tells you, I'm going to say semi-quanta, oh gosh, I can't even spell it. Quantite, oh geez, Louise, that was bad, wasn't it? Semi-quantitative. So you kind of get an idea of how much sugar is present. Anywho, that's going to get you all the marks. Now we're on to question three. So let's zing. And question three runs like this. Fermentation in yeast is used in the manufacture of bread and alcoholic drinks. The equation for fermentation is glucose goes to ethanol and carbon dioxide. Fermentation is an exothermic reaction. What does exothermic mean? So what do you think exo means? Exo, like, is exit, going out. Exothermic is heat is released. Heat is released. Respiration is an exothermic reaction, so is fermentation. You can put your hand under your armpit, you can feel heat coming out your armpits right now, and that's an exothermic reaction. That's the heat from respiration. Fermentation is also a heat producing reaction, so heat is released. Opposite of photosynthesis, which is an endothermic reaction, so takes in heat. So if you have lots of plants in your living room, it will have a cooling effect in the summer because they're going to absorb heat from the room. Okay, a student investigated the effect of temperature on the fermentation in yeast. Figure 3 shows the apparatus. We've got a gas syringe. Should have had that in the photosynthesis experiment. We've got a water bath. Look, they've got a thermometer to measure the temperature. We've got a layer of oil here, and that is to control the temperature. We've got a layer of oil and yeast in the sugar. We've got a flask. I bet they're going to ask us what is the oil for, but let's have a look. This is the method. Mix the yeast with sugar solution in a flask, pour a layer of oil over the surface of the mixture, put the flask in a water bath at 2 degrees and leave for 20 minutes. Attach a gas syringe. Record the volume of gas collected every 5 minutes for 30 minutes. After 30 minutes, move the flask to a water bath at 35 degrees. Continue to record the volume of gas collected every 5 minutes. Suggest why a layer of oil was needed on the surface of the mixture. So what they're doing here, and I'll just go back and see if we can get a clue from up here. Student investigated fermentation. Let's find out a bit more. Fermentation in yeast is used to manufacture of bread and alcohol, the equation. What's missing from this equation? It's not aerobic respiration. Got sugar, there's no oxygen. So how do they make sure the yeast are fermenting the glucose? They don't want oxygen because then they won't produce that ethanol. So it suggests why a layer of oil was needed. It's to prevent can't actually spell, can I? To prevent oxygen to entering, whoops, entering, oh my gosh, this is coming out. Helios. To prevent what the fudge? To prevent oxygen entering the flask. Sugar Ray Leonard, this is awkward. You could say to make sure it respires anaerobically because fermentation is ethanolic or alcoholic fermentation like this is an anaerobic process. So we want to ensure that the yeast does anaerobic and doesn't switch to aerobic. Okay, suggest why the mixture was left for 20 minutes before the gas syringe was attached. So think about it, you've just bung the yeast in there. Why leave it for 20 minutes before you start collecting gas? We want the yeast, all their enzymes and everything to warm up and to get to the correct temperature. We kind of say equilibrate or to acclimate so the yeast can acclimatize. We've just mixed it all together. The yeast might be at 10 degrees. You've got it out the fridge. The oil might be at 12 degrees. The water that you're putting in there or what else might be at another temperature. And you've set the water bath at 20 degrees. You want it all to get to the same temperature before you start recording. And you want it all to acclimatize the yeast to get used to doing their respiration thing. So the yeast can acclimatize to the environment. So the temperature is equal in the flask contents. You know what I mean? So the heat or the temperature is spread evenly in the flask contents. Anywho, that was all a bit long. That was a suggest. So suggests are always like you can waffle a little bit in a suggest as long as it's sensible. Right, steps 1 to 4. Um, Step 1 to 4 of the method was repeated at 35 degrees. The volume of gas collected was recorded every 5 minutes for 45 minutes. Table 2 shows the results for both flasks for the first 30 minutes. Table 3 shows the results for the last 15 minutes when both flasks were at 35 degrees. So we've got table 2, we've got the time, and then we've got the flask at 2 degrees. And that's the volume of gas collected when it was at 2 degrees. Look, absolutely nothing. And then the flask at 35. So as the time is going up, it is collecting more gas, although it does sort of slow down after a while. And then here is volume of gas collected in centimeter cube, time in minutes. Flask 2 was then moved to 35, and you can see it started to increase. Um, the gas is being produced basically. And then the flask at 35 hasn't increased anymore. So we're going to be asked about what's going on in these tables. Explain the results from 0 to 45 minutes for the flask that was 2 degrees and then was moved to 35. So let's have a little look and see what's going on. So when it was 2 degrees, why was there no gas collected? What doesn't work well at cold temperatures in biology? What things that control all reactions and they're inside every living thing do not work well at cold temperatures? The answer is enzymes. So at 2 degrees, the enzymes do not have the kinetic energy to collide with the substrate. Their active site doesn't bind with the substrate. They don't catalyze the reaction. It's too slow for enzymes to work. Too little kinetic energy, too few collisions. But then when we went to 35 degrees, we now have more energy, more kinetic energy, more collisions between the substrate and active site. So we have an increase in the rate of reaction and more gas. So I just got to type all that down. Geez, Louise, lucky me. So at 2 degrees, no gas was produced as the enzymes did not have enough kinetic energy. Um, no collisions between. How do I know there were no collisions? Because there was zero gas collected, so no collisions between active site and substrate. Um, so no reaction. So there was no reaction going on. That has explained the 2 degree phenomenon. Now, you can probably say 2 for each and get 2 marks for each, but it's a maximum of 3 marks. So I can just say one thing if I want for at 35. So at 35 degrees, but I'm going to say a bit more. More kinetic energy, and we want to say more collisions between active site and substrate. And 35 degrees, I could say 35 degrees is closer to the optimum temperature for enzymes. So close to that 40 degrees or whatever, 37 or whatever yeast enzymes their optimum temperature is. So there we go. Now, oh, Sugar Ray Leonard, I put a sugar tip. Hey, hey, hey. 3.5. Explain the results from 0 to 45 for the flask kept at 35. Oh my good gosh, please. Right. So at 35, we've got the same business going on, right? So what we can see here is 35 degree, it's close to the optimum temperature, lots of kinetic energy, lots of collisions between the active site and the substrate. So we've got lots of gas being produced. But when it gets to the highest rate of reaction here, 0 to 26. So in the first 5 minutes, we've got the steepest rate of reaction. And then all the way up to 10 minutes, it's reacting very quickly because there's the most substrate and active sites. However, what's happening now, we're running out of substrate, substrate's being broken down, we're running out of reactants, and eventually we get to the point where there's no reactants left. So there's the enzymes are just sitting there with nothing to break down. So we've got no more gas being given off, even though the temperature is still juicy and there's lots of kinetic energy. All right. let's get that stuff down. I probably won't get it down as well as I just said it there, but there we go. So, um, high rate of reaction during first 10 minutes as lots of substrate slash reactants um, to collide with active sites of enzymes. However, um, whoopsie, what's the temperature? I've totally forgotten what it reached, guys, which is a pain in the backside because you probably have to quote the number, but I'm not going to flick back up. Not the temperature. However, I'm just going to say eventually, but you would need to actually say from the graph or the table what time this took place. So, however, I think 5 minutes, it was 40, 35, 30. However, eventually, after 30 minutes, there was little increase in gas produced as all the reactants or substrate had been used up. Um, so not a lot of collisions between active site and substrate. Okay, I've kind of answered this in terms of enzymes. Anywho, I will check the mark scheme and then add that in later, maybe when I upload a video. Gosh, we are on question 4. Okay, so number 4, pathogens are microorganisms that cause diseases. Gonorrhea, malaria, and measles are 3 diseases in humans. 4.1, draw one line from each disease to the pathogen that causes the disease. So we've got gonorrhea. What causes gonorrhea? Well, gonorrhea is caused by a bacteria, and that will get you a mark. Gonorrhea, by the way, you get green or yellow pus from your genitals. Not nice. Pain when urinating. So not nice. And it's a sexually transmitted disease. Now, malaria is caused by a protist, the Plasmodium protist that's carried by mosquitoes, which are a vector. And you need to know all of that. I'm missing something here, aren't I? Yeah, you donkey Tom. Sounded like Donkey Kong. You donkey Tom. Right. So that goes there. That goes there. Gosh. Gosh. Measles. Yep. And look there. Measles are caused by a virus, coughing, droplets. They give you a fever, but you're mostly vaccinated against measles for protection. There you go. 3 marks. Bish, bash, bosh. Right. 4.2. Malaria is transmitted by mosquitoes. Male mosquitoes can be sterilized, so they are infertile. The spread of malaria is reduced by releasing sterile mosquitoes into the environment. Explain how releasing sterile mosquitoes reduces the spread of malaria. So I'm going to give you the colloquial or the slang version of it. Basically, it's getting the mosquitoes and you're radiating, firing irradiation, gamma, alpha, beta, you know the stuff, ionizing radiation onto the testes. Well, I don't know if mosquitoes have testicles, but you're killing their sperm. Now, the mosquitoes then go out and have sexual intercourse with the females. The females think, oh, great, I'm pregnant. No need to go and sleep with any other mosquitoes. I'm pregnant. And of course, they're not really going to be pregnant because the male mosquito's sperm is sterile. So for that whole generation, that whole breeding cycle, the females don't have any offspring, and therefore the population has a massive dip. And it's much better than going around trying to catch the male mosquitoes and kill them. If you only kill 80% of the male mosquitoes, you're going to leave 20% alive, and they're going to go and have sexual reproduction, successful sexual reproduction with all the female mosquitoes, and you're not going to affect the population. Whereas if you sterilize a load of the men, they will go out and have sex with the females, and the females will think they're pregnant and won't bother sleeping with any of the men that are still fertile. And there we go. I've just got to put that in words. Explain how releasing sterile mosquitoes. So we want to say kills the sperm um, of the males um, when they reproduce. The females cannot get pregnant, or yeah, do not produce offspring Uh, decrease, I'm sure you could just write decrease in population. This is too long. Decrease in population of mosquitoes. Um, so less people bitten by mosquitoes. Yeah. So you get less spread of the disease because there's less mosquitoes because there's a decrease in the population. Mosquitoes carry malaria, obviously. They carry that Plasmodium protist. It hides inside them and waits till they suck on the blood of someone, swims down their little proboscis and into the person. Okay, pathogens also cause diseases in plants. Figure 4 shows a rose black spot fungal spore and a tobacco mosaic virus. There's the tobacco mosaic virus. Look how small it is, 2.5 times 10 to the minus 7. And the rose black fungal spore is 16 micrometers. Images are not to the same scale. Yeah, we don't need that. We've got the numbers. Name the piece of equipment used to view the virus. What would you need to view something that small? I know everyone's screaming, microscope. What type of microscope? You couldn't see a virus with the microscope you have in school. You'd need this one. You'd need an electron microscope. Now, um, the virus you've got in school could see a fungal spore and it could see a bacteria, but it couldn't see a microscope. Okay. It couldn't see a virus, it could see a microscope. Yeah, right. Move on. Mm. Electron microscope would set your school back about 100,000 or 200,000 pounds. So don't think your school's being tight. 4.4. How many times longer is the fungal spore than the virus? 3 marks. What we got to do here, peeps. This is in meters. This is in micrometers. First things first, we need to convert them both into the same unit. So let's do that first. So if I want to convert this into micrometers, I could first convert it into millimeters and then into micrometers. So I could times by 1,000. There are 1,000 millimeters in a meter. So if I go 2.5 times 10 to the minus 7, right? And then if I times that by 1,000, I'm going to get 2.5 times 10 to the minus 4, and that's millimeters. I'm still not at micrometers. I need to times by 1,000 again, and that is going to get me into micrometers. Um, so, and I'm going to get, if I times by 1,000 again, we're going to get 0.25 micrometers. We've now got the same units because I put the virus into the same units. And now I can see how many times bigger is that. And of course, 16 over 0.25 is the same as saying 16 times 4. So what's that? 44, 60, 64, I believe. Ciao. And there we go. That's going to get you 3 fat marks. 1 mark for converting into micrometers, 1 mark for dividing, 1 mark for the answer. Bish, bash, bosh. Let's go. All right, I'm not going to say bish, bash, bosh any more times in this. So that's getting seriously annoying, and that's getting annoying for me. So 4.5, explain why plants infected with the tobacco mosaic virus grow slowly. So anything that infects or diseases plants, it's the same blooming answer every single time, okay? It's the same flaming answer. So we're just going to say it can't do as much photosynthesis. Therefore, it can't make as much glucose. Therefore, it doesn't have the energy from respiration to grow. So it doesn't matter what virus, what it is that damages a plant. The answer tends to always be the same with plants. If you're going to mess plants up, it's because you've messed up photosynthesis. So what we can say is the leaves are damaged or discolored. There's a bunch of things we can say here. Leaves damaged and discolored because the leaves go funny with tobacco mosaic virus. They get that patchwork, hence the name mosaic. We could say, if we really want to get funky, less chlorophyll in leaves. And if you have less chlorophyll, you're going to have less photosynthesis. That means less glucose produced. And if we have less glucose, that means less respiration, less energy for growth. We could also go down the line of less glucose means less amino acids, less proteins that are needed. You know, proteins are for growth and repair that are needed for growth. So that's why plants that have the tobacco mosaic virus will grow slowly because they don't have a lot of energy and they can't make protein, which we need for growth. There's about like 50 marks here. Okay, awesome. Okay, we're on to question 5. Who is the heart? Figure 5 shows the human heart. We've got A, B, C, D. Which blood vessel transports blood with the highest oxygen concentration into the heart? So let's go. We can either be A, B, C, or D. So we want blood with the highest oxygen concentration into the heart. So I'm going to go through this. Blue blood returns to the heart via the vena cava into the right atrium, down into the right ventricle. This is deoxygenated, always enters through the right side of the heart, deoxygenated blood. So that's not going to have a high oxygen concentration, is it? But A does go into the heart at least, so part of it's right, but it's definitely not going to be very high. B is leaving the heart, going up to our lovely lungs. And then what happens at the old lungs? Well, it goes into capillaries and runs through the alveoli, and then it comes back to the heart. That's the pulmonary artery, by the way. Artery, the A in artery means away from the heart, and then it comes back through the pulmonary vein into the left atrium, left ventricle, pumped up through C, which is the aorta, and off it goes round to the body. So this question said, which blood vessel transports blood with the highest oxygen into the heart? There's only two things going into the heart. It's here at A and here at D. But the one with the highest oxygen will be D because it's just come back from the lungs and picked up a load of oxygen. So there we go. 5.2, blood pressure is a measure of the force of the blood against the walls of the blood vessels. Which blood vessel transports blood at the highest pressure? A, B, C, or D? Well, we can check this out. So I just slapped that down there. Oh my days. What have I done there? Okay, think about it. Which side has got more muscle? Which side is going to be pumping the hardest? It's not A, it's not this chamber or this chamber, the atria. They're only pumping down to the ventricles. This ventricle only goes like 15 centimeters to the lungs. This one is going meters around the body. So this blood vessel is carrying blood at the highest pressure, C, because this left side of the heart with all that big fat muscle is having to contract with a lot of force and create a high pressure. So which blood vessel? The answer is C, has blood at the highest pressure. That's the aorta. It's going to be the highest pressure in the whole body. 5.3, what is the correct order for blood flowing through the heart to the lungs? Oh my good gosh. So we've got left atrium to the left ventricle to the pulmonary artery. Um, we've got left atrium to the left ventricle to the pulmonary vein. We've got right atrium to the right ventricle to the pulmonary artery. And that is the way we go from the heart to the lungs. So once again, going back up to my little rusty diagram, we're we're coming in here through the right atrium, right ventricle, up through the pulmonary artery to the lungs. So there we go. It is going to be the third row down, and I'll get you a mark. Best way to learn the heart, get a scrap bit of paper, sit down, watch something on TV, try and draw the heart out and label it all. And that's a good way to revise when you're just not in the mood for answering questions or doing some deep reading. Every year, thousands of people in the UK have heart attacks. A heart attack is caused when the heart muscle cells do not get enough oxygen, causing the cells to die. Statins and stents are two treatments used to reduce the risk of someone having a heart attack. Evaluate. Now, evaluate means say the positive, say the negative, and then give your opinion. You have to give your final conclusion, your summing up, to get that sixth mark. Give your opinion. So say the good, say the bad. Evaluate the use of statins compared with the use of stents to reduce the risk of a heart attack. So we want to evaluate. So both reduce the risk of heart attack by increasing blood flow to the heart. Okay, so they both do the same thing, which is reduce the risk of heart attack by increasing blood flow to the heart. However, stents require an operation. So stents require an operation, and what does that bring with it? There is a risk of infection. So when you have an operation to fit a stent, you could have an infection, so that's a risk. Also, you may get blood clots at the stent site. So that's a problem in itself because the stent's a piece of metal. It could produce a blood clot at the stent site, and that could give you a risk of heart attack. Operations are always a little bit risky. whereas statins are medicines, stroke drugs, um, do not require operation. However, statins can have side effects. So there are like simple side effects from statins, such as um, headaches, but serious, rare side effects. I've not worded this very well, but serious, rare side effects, such as organ failure. And of course, that can lead to death. So statins could potentially kill you. Also, you have to remember to take statins every day. So there's a little bit of an issue there. Like just because you're on statins and you haven't got to have an invasive procedure like an operation, you still got to remember to take them every day. But how do statins work? So they work by two. Both of them have two different mechanisms that increase the blood flow. So what we would want, we've really talked about all the negatives about them, but of course, um, there are good things about them as well. I've just kind of mainly focused on the negatives here, but how stents require an operation, there's a risk of infection as you may get blood clots, but they're not drugs. You don't have to take them every day. Statins are medicines. They do not require the operation. However, they have side effects, could be serious, and you have to remember to take them every day. You could have an allergic reaction to the side effects. Yeah, we said that you could get a blood clot with the statins. I probably should add in this could lead to a heart attack. Might as well say what the blood clot's bad for. And then I should really say stents are good as they open up the artery, allowing increased blood flow. Okay. Whereas statins work in a slightly different way. So I should really get down how they work. Statins reduce cholesterol and so reduce buildup of fatty deposits. And that makes you much less likely to have a thrombosis and a heart attack. But they both kind of increase blood flow by those methods. So there we go. Loads of arguments for and against. Personally, I would definitely finish with an argument saying which one you prefer. With evaluate, I always like to give my opinion. So you'd then give your opinion. You can choose either one and do a final sentence just to say, listen, save something for one of them. So maybe say like overall, statins are better because it's cheaper for the NHS because the operation is more expensive. So you could say something like that. Or you could say stents are better because the stent will remain in place for a long time and the effect of the stent is immediate, something like that. Who knows? The operation doesn't last long, quick recovery. Anything nice and positive to say about the stent? Right, we're on to the nearly on to the final question. Jeez Louise, this is going on and on and on. Many people who survive a heart attack get out of breath easily when they exercise gently. Explain why heart attack survivors get out of breath easily. So this is going to be, if you're a heart attack survivor, the heart cells have died. So heart cells have died. So the muscle cannot contract as effectively or forcefully. So it doesn't contract with the same amount of force. It's gone weak because quite a lot of the muscle has died. This means it's the old, this is so common in any heart problem. You put this next little spit. So this means less blood flow. We go less oxygen to body cells, less respiration. I should really say aerobic here because oxygen is required for aerobic. So less respiration, aerobic, less energy released. I shouldn't say made, I should say released, less energy released. So feel tired. Now, we could also say more anaerobic, because if you don't have oxygen, you're going to do more anaerobic respiration, more lactic acid, which causes fatigue. So lactic acid itself is a toxin and it causes muscle soreness and fatigue. So there's tons of marks here, guys. But that is always very common. This little bit here that I've said here is very, very common when you're doing heart content. That little flow of less oxygen, less respiration, so less energy released, more anaerobic respiration because you can't do aerobic respiration. That is very, very common. Okay, scientists have developed patches of beating heart cells to repair damaged heart tissue. The patches are placed on areas of the heart where the cells have died. New cells grow to replace the dead cells. The patches are made using a person's own cells that are converted into stem cells. Explain why stem cells are used to make the patches. So what we want to say about stem cells, think about the keywords linked with stem cells. Stem cells are undifferentiated cells. You can say unspecialized cells, or you can say they're not differentiated or not specialized. Um, this means they can become any type of cell. They can specialize and become any type of specialized cell, and that is going to get some marks. So we've said what stem cells are and what they can do. The scientists could have used human embryonic stem cells to make the patches, give two advantages of using stem cells made from the person's own cells rather than using embryonic stem cells. So, what are the advantages of using your own stem cells? Okay, well, if you use your own stem cells, we can say no embryo is destroyed. Um, And the second advantage is if we use your own stem cells, they will contain the same antigens, the protein markers on the outside, so they'll be recognized the same and they won't be rejected, which means attacked by your own white blood cells. So less chance of rejection. Now, rejection just means that your white blood cells, when something is rejected, your white blood cells attack it and destroy it. Um, And so that's no good if you get some stem cells to grow you something new and your own white blood cells attack parts of your own body and destroy it. We don't want that. That's what rejection is. Okay, this question is about plant transport systems. 6.1, describe how water is transported from the soil to the atmosphere through a plant. So what we're going to look at here, you have the roots of a plant, and then we have the stem of a plant, and then we have the leaves of a plant. And what we're looking at is the journey that the water goes on, and that's what we're talking about. So first of all, how does the water, the H2O, get into the roots? Then how does it move up the stem? And then how does it get out of the plant? And this whole process has a name, and a lot of people forget that this is a different process to one of the others. I'll point this out anyway. I'm going to say water um, travels through the transpiration stream. Now, that is a different thing to just saying transpiration. And in itself, this might be worth 2 of the 4 marks, just saying those 2 things. So the transpiration stream, I'm going to say water enters the roots via osmosis. So water goes into the plants from the soil into the roots via osmosis from a higher water potential, or we can say concentration in the soil, to a lower concentration in the roots. Okay. Now it travels up the stem. You don't need to know at GCSE how it goes up the stem. So we're just going to say it travels up the xylem. You don't need to know how it goes against gravity. It's by a process that's not done at GCSE. So it travels up the xylem. So any mention of the water moving through the xylem is another mark. And then it evaporates out of the stomata in the leaves by transpiration. Now, you might say, what's the difference between transpiration and transpiration stream? So I just want to highlight that because that's come up before in other tests. So the word transpiration and transpiration stream are linked, but they don't mean the same thing. So the transpiration stream, it is basically describing, is a term used to say water goes in by osmosis up the xylem into the leaf and evaporates out of the leaf. So it's the whole thing. It's called the stream or the river of transpiration. Call it the transpiration stream. Whereas transpiration, if you just use the word transpiration, you're just referring to this bit when the water evaporates out of the stomata on the underside of the leaf. Okay, so that is what just using transpiration means. So it's best to say the whole thing, transpiration stream. There's 4 marks. Lovely jubbly. Right, 6.2. Dissolved sugars are moved through a plant in a phloem tissue, what is the name of the process that moves dissolved sugars through the phloem tissues? So you should know this. It's not transpiration, but it sounds very similar. It's translocation. And that is going to get you the mark. Okay, let's zing, zing, zing down to the next question. We're on 6.3 now. Phloem tissue is made of sieve tubes and companion cells. This is the phloem tissue. Figure 6 shows a section of phloem tissue. We can see the companion cell has mitochondria and a nucleus. The sieve tube cell doesn't have any of those things inside it. Okay, but it has these weird gaps at the end in the cell wall. Look, these little gaps look like a cheese grater. Explain one way sieve tubes are specialized for their function. So we've just kind of discussed how the sieve tube cells could be specialized for their function. We only have to name one way. So I talked about the cheese grater effect. They have holes in the end wall to allow small dissolved substances to pass. So if they didn't have holes, things wouldn't be able to get through here. So that's why they have these holes here. And that get me 2 marks. If you say there's some holes in the end of the wall to allow small dissolved substances to pass, if you want to give an example, glucose or amino acids are things that move around inside the phloem. Something else we could say for 2 marks, another double bubble, would be there is no nucleus. There's also no other organelles, no mitochondria or any of that. So we could say that there is no nucleus. This makes it easier for substances to flow. There's less resistance to the movement of substances. Sorry, my phone is blowing up, so it's like beep, 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 beep. Yeah. All right, enough of that waffle. No, that's not working. That's my boss from work texting me at 8:30 at night. What a gift. Right, 6.4. What does the structure of the companion cell suggest about the process that moves dissolved sugars through the phloem tissue? So why we've got a companion cell there. What does that tell us about the process? Okay, you might know the name of the process, but what is the fact that you need this? They've been very clear. Look, they haven't done this by mistake. They haven't shown ribosomes. They haven't talked about anything else. They've labeled the mitochondria. So what does that tell us about the way that substances are moved in the companion cell? What does that tell us about the way they're moved? So hopefully you might clock, what is the purpose of mitochondria? What do they do? So they have mitochondria, which do aerobic, no, aerobic respiration, um, release energy. What process requires energy? Is it diffusion? Is it osmosis? 10 points. It's active transport. For active transport. So translocation is an active process which involves active transport, hence the need for all those mitochondria, and hence why they're in the companion cell. If the mitochondria were in the sieve tube, they'd get in the way of the flow of stuff. Yeah, it'd be like hurdles in the middle of the racetrack. Okay, final question, 6.5. Describe why it's important that dissolved sugars are moved both upwards and downwards in a plant. So think about a plant. You know, it's got roots, it's got a stem, it's got leaves. If you're making sugar in the leaves, why might you want it to move higher, the sugar? And why might you want it to move down lower? So it's really simple. We might want to move sugar down towards the roots. What for? So sugar is moved down to the roots for storage as starch. You know, roots, potatoes, carrots, rice, all of that kind of stuff is very starchy. What about also for respiration in the roots? The roots also do active transport, also for active transport of mineral ions. So the roots need to actively transport nitrates in. So they're going to need sugar to do respiration to get energy for the active transport of mineral ions. So I'm kind of linking all that to the leaves for respiration for growth of new leaves. Okay, there's loads we could go on about. We could talk about all sorts. Basically, respiration in the roots, respiration in the leaves. Why in the roots? You might need energy for active transport and for the growth of roots, and you might need energy for the growth of new leaves. And that's why we move it around. And we also might want to move sugar down to the roots to be stored as starch. So lots and lots of reasons why that might happen. There we go. That's the end of the test. Share and scare and hit the like, punch up the bell, subscribe. Let's get to the marks.
You still here? It's over.